Test. Good? Okay. According to 2020 data from the CDC, 1 in every 3 adults and 1 in every 5 children in the United States were diagnosed with clinical obesity, encompassing more than half of our population. With the additional inclusion of overweight individuals, there is a big concern for obesity-associated conditions. Several health conditions are associated with obesity and increased fat mass, including cancer. The risk of getting cancer is increased, but the odds of responding to treatment and surviving long-term are decreased in these individuals. Public narrative around this problem of obesity-associated conditions has long been that individuals should just lose the weight, but we know that is not a readily available solution for everyone. We believe that people deserve to be effectively treated where they are, rather than where we want them to be. In our lab, we studied the obesity-cancer relationship in an acute form of B-cell leukemia. This leukemia is the single most common childhood cancer and is a leading cause of illness-related death in children. These leukemia cells form in the bone marrow and are surrounded by other cell types, such as adipocytes or fat cells, shown here in yellow. The bone marrow also houses immune cells known as killer T cells, whose job is to find and protect the body against foreign invaders, such as cancer. Ideally, these T cells will find the leukemia cells and destroy them, curing the person. However, this process is imperfect and is inhibited when bone marrow adipocytes are increased. To study this process in the lab, we use a culture of adipocytes to see what factors they are releasing that are interacting with these other cells in the bone marrow. These adipocytes release many different things into their local environment, and we collect them by growing adipocytes in their favorite nutrient bath and then collecting their bath water after several days. When we measure the different factors that are present in this bath water, we can understand what the adipocytes are releasing into their local environment. One that we found particularly interesting is a small molecule called guanine, which I am focusing on for my dissertation. When leukemia cells are grown in the presence of guanine, their cell cycle changes. The cell cycle is the process by which cells grow and is also a key target of many cancer-killing chemotherapies, meaning that the presence of guanine may prevent leukemia cells from responding to chemotherapy treatment. Guanine also interacts with the T cells and inhibits their ability to find and kill the cancer cells. This means that guanine released from adipocytes may both prevent leukemia cells from responding to chemotherapy treatment and prevent the cells from being targeted by cancer-destroying immune cells. Now that my research has helped identify a factor that may be responsible for the worst outcomes in cancer associated with obesity, we can start developing methods to block guanine and improve outcomes in these particular patients. Targeting guanine will enable us to improve therapy outcomes for individuals with leukemia and patients of all shapes and sizes. Thank you.